Dear students, in my last class, I gave an introduction on cell signaling process. Now, today I will focus on one of the signal receptor molecule, or we can say which is present on the cell surface of the target cell. This receptor is known as tyrosine kinase receptors, and it is involved in signal perception stage of cell signaling in the target cell. In last class, I told you that when a ligand molecule, which is commonly known as signal molecule, either by paracrine signaling, endocrine signaling, autocrine signaling, juxtacrine signaling, it binds to the receptor molecule of the target cell. The receptor molecule may be present on the surface of the cell. When receptor molecule is present on the surface of the cell, the receptor will be called as cell surface receptor. And when the receptor molecule is present inside the cell, then the receptor molecule is called as intracellular receptor. So by this we can categorize this receptor molecules. The common cell surface receptors they can be also called as plasma membrane bound receptors. The common type of receptors are tyrosine kinase receptor, G protein linked receptor, and ion channel linked receptor. Tyrosine kinase receptors, G protein linked receptors, or ion channel linked receptors are those receptors which bound to ligand molecules, those ligand molecules which cannot cross the membrane. It means these ligand molecules are either charged molecules or they are polar molecules. Like they may be proteins, they may be biological amines, they may be even ions also. So they cannot cross membrane. While the intracellular receptors, on intracellular receptors, ligand molecules which are of lipid origin, they bound to intracellular receptors. Like many hormones, progesterone, estrogen, testosterone, they all are lipid in origin, so they bound to intracellular receptors. So today in this lecture I will focus on tyrosine kinase receptors. Tyrosine kinase receptors are also known as enzyme linked receptors. They are so named because their cytosolic domain behave as enzyme. Now, in last class I had already mentioned you that all the plasma membrane bound receptors or cell surface receptors are transmembrane proteins. Transmembrane proteins are those proteins which flanks the lipid bilayers. They have an cytosolic domain which is inside the cell. They possess extracellular domain which is outside, which is facing outside the cell. We are towards extracellular matrix. So in this picture you can see on the left side the single pass integral protein. You can see the extracellular domain on the upside and the cytosolic domain on the downside, and in between the lipid bilayer there is a region of protein which is called as alpha helix, and this alpha helix is spanning the membrane only single time, so it is called as single alpha helix membrane spanning protein. Tyrosine kinase receptor is actually a single alpha helix membrane spanning protein, just like in this picture. The N-terminal domain is facing outside the cell The C-terminal domain is facing inside the cell, and the part of the polypeptide which is present inside the lipid bilayer is always rich in non-polar residues. Non-polar residues like glycine, alanine, valine, methionine, leucine, isoleucine. These amino acids are non-polar in nature, so they can easily be present. They can easily bound with accommodate inside the non-polar nature of the lipid molecules. That is the triacyl glycerol. While the polar residues are found either outside or inside. So the inside one, the C-terminal domain of the tyrosine kinase receptor actually act as an enzyme. So therefore, it is also known as enzyme linked receptor. In green color, you can see a protein molecule. It is also a transmembrane protein. You can see, but more than one alpha helix present inside the hydrophobic core of the lipid bilayer. So when more than one alpha helix is present, so they can be called as multiple alpha helix membrane spanning protein. For example, G protein linked receptor is actually a multiple alpha helix membrane spanning protein, and it has 7 alpha helix, similar to bacterial reduction. But today in this lecture, I will focus only on tyrosine kinase receptor, the structure, how it gets dimerized, how it gets activated, how it transmit the signal inside, what type of signal transmission pathways operates on activation of tyrosine kinase receptor, and what type of cellular responses it generates in the target. So in this picture you can see tyrosine kinase receptor in A picture and tyrosine kinase receptor in B picture. Actually, tyrosine kinase receptors are present in in dimeric form, means they are two different polypeptides, and each polypeptide is a single alpha helix membrane spanning protein. So they may be present in inactivated state, or they may be present in activated state. During inactivated state, tyrosine kinase receptors are never present in dimeric form. They are present far apart. And one important thing is about tyrosine kinase receptor is that their cytosolic domain is rich in tyrosine amino acid. That's why they are known as tyrosine kinase receptor. Tyrosine amino acid is supposed to be phosphorylated during activation, and when can see it is an overview of TKR molecule, how it generates a protein kinase cascade. So the signal molecule bind to the TKR, the receptor activates a molecule. This molecule again acts as a kinase enzyme. It activates inactive protein kinase 1, then inactive protein kinase 1 is activated into active protein kinase 1. It activates another protein kinase, and so 2, the protein kinase 2 is activated, then protein kinase 2 acts on its substrate, which is protein kinase 3. So in this manner, in this cascade manner, each protein kinase activates another protein kinase, and that protein kinase activates another protein kinase. So a cascade of protein kinases is being generated during tyrosine kinase receptor during signal transmission pathway. So finally, a protein kinase cascade can activate any inactive protein to active protein, or any inactive enzyme to active enzyme, or any inactive transcription factor into active transcription factor. That active transcription factor may bind to either enhancer or silencer of the gene and can regulate the gene activity. Any enzyme which is activated or inactivated, it can catalyze reaction or can stop catalyzing reaction. So these all will be called as cellular responses. Now MAP kinase cascade is a common protein kinase cascade. This protein kinase cascade is also known as protein serine or threonine kinases, because the protein kinases present in signal transition pathway are rich in serine or threonine residues. You can see in the MAP kinase cascade. MAP kinase cascade is initiated by binding of a growth factor. Growth factors are actually the molecules or the signal molecules which are responsible for initiating cell division in the target cell. There are various different types of growth factors like EGF, epidermal growth factors, IGF, insulin-like growth factors. So all these growth factors bounce to the target cell by tyrosine kinase receptors, and then initiate their signal, and they then pass their signal to the target cell. The target cell gives response by division of the cell, but this cell division is tightly controlled. If any mutation occurs in signal transition pathway, it would be lethal. Now what happens in MAP kinase pathway? MAP kinase means mitogen activated protein kinase. Mitogen means the substance which induces mitosis. So here the ligand molecule, the growth factor is a mitogen. So mitogens and this mitogen activates the protein kinase cascade. Therefore, it is called as popularly known as MAP kinase cascade. So when GEF bind to receptor tyrosine kinase, it gets dimerized and get activated. It activates RAS protein. Now RAS and RAF protein are very important, and it was found to be that RAS protein is found to be mutated in 30% of human cancer. So it is very important protein because it is part of signal transmission pathway. So RAS activates the RAF protein, and RAF protein activates initiate a sort of protein kinase cascade in the cytoplasm, which is called as MAP kinase. And ultimately, they leads to activation of transcription factors which are responsible for expression of enzymes like or expression of genes like G1 cyclins, which are responsible for crossing the cell, passing the cell from G1 phase to S phase, that is at G1 checkpoint. They are responsible for expression of enzymatic machinery which are related with the DNA replication, means the expression of DNA polymerase and subunits, expression of helicases, expression of topoisomerases, expression of single stranded binding protein. Means if such type of proteins and enzymes is being synthesizing inside the cell, it means the cell is preparing for DNA replication, and when cell is preparing for DNA replication, means cell is going to be divided. Further, this is the cellular response inside the nucleus, but they also reorganize the cytoskeleton structure and many other enzymes in the cytoplasm as well. And the cellular response is what? Division of cell. Thank you.